Have you ever wondered why the same treatment can work for one patient but not for another? Same diagnosis, same medication, treated by the same doctor in the same hospital, but after 2 weeks, different response. In schizophrenia, is a real clinical challenge. We know that atypical antipsychotics are a mainstay therapy for schizophrenia, and they work by interacting with receptor. Think a receptor as a lock and the drug as a key. One important lock is serotonin to a receptor encoded by STR to a GAN and targeted by both of risperidone and clozapine. So, what lies behind in this difference? Part of the answer be writing in our genes? To answer this question, we studied 160 Batak patients with schizophrenia, all receiving combination therapy, risperidone, and clozapine. We know that Indonesia is genetically diverse, but the Batak population has a relative homogeneous genetic background, making it easier to study pharmacogenetic variation. Treatment response was assessed by using PANSS, with responder showing at least 20% reduction in total PANSS score. Then we looked into their DNA, focusing on two variants of STR2A, RS6311 and RS6313. What we discovered was even more interesting. Among all inheritance models, the recessive model showed the strongest association with the treatment response. Patients having two copy of risk allele were more likely to be non-responder to risperidone and clozapine therapy. But the evidence didn't stop there. When we look at the combination of these two variants, even stronger evidence emerged from the haplotype analysis. The TA haplotype was associated with the better response, and whereas the CG haplotype was associated with the poor response. Although in this study we didn't measure directly about the functional of the receptor, our findings suggest that STR2A genetic variation may contribute to difference in antipsychotic response. So remember the two patients at the beginning, same diagnosis, same treatment, different response. Our findings suggest that part of the reason may be written in their DNA. Ultimately, this study may bring us one step closer to personalized treatment in schizophrenia. Perhaps in the future, we may no longer ask, what should we prescribe first for this patient? Instead, we may ask, which drug is most likely to work for this patient based on their DNA. Thank you.